We are going to start now. If you can please take your seats. You can bring your coffees to the seats.
Okay, if we could ask everybody to be seated. We're going to finish our last session this afternoon of the Science Cafe. So we're moving closer to home. Xian Ying is a junior principal investigator at IMCB here in Singapore. And she's an adjunct assistant professor at NUS. She came back just a couple of years ago, even less, yeah? Well, about a couple of years ago, where she trained with Lee Rubin at Harvard. And she's going to tell us a little bit about some of the work in her group on patient-derived iPS cells, in particularly the work that she's been doing in ALS-derived iPS cells to identify a novel target. Is this working? Can you hear?
Thank you, Andy, so much for the introduction. So my name is Xian Ng. I'll be talking a little bit about our work on using patient-derived induced pluripotent stem cells to understand neurodegeneration. I'd like to start by giving a brief background of myself. I did my PhD here in Singapore at the Genome Institute of Singapore, and that scholarship was sponsored by NUS. I then went on to Harvard to perform postdoctoral training under Lee Rubin's lab, and then came back as a principal investigator at the IMCB here in Biopolis. At the same time, I also have an adjunct assistant professorship at the National University of Singapore. So my research interest involves using iPSCs, or induced pluripotent stem cells, I will talk more about them in subsequent slides, to model neurological diseases. And importantly, we would want to identify genetic and/or signaling pathways that are dysregulated in disease, hoping to eventually target them for therapy using small molecules. So what is amyotrophic lateral sclerosis, or ALS? It is essentially an age-onset motor neuron disease. Your risk of getting ALS gets larger as you get older. It affects almost 500,000 people worldwide, with 30,000 in the USA alone, and 5,000 new cases every year. In the local context, about 300 to 400 people in Singapore will get the disease. So ALS can be caused by several different mutations in different genes, and this involves SOD1, TDP43, or a gene called FUS or TLS. It can also happen in the absence of known mutations, and this is what we call sporadic ALS. Most people would die between 3 to 5 years from when symptoms first appear, and these symptoms can include general things like loss of muscle tone or muscle weakness. And unfortunately, there is no cure for ALS at the moment. The only options patients would have is a drug known as riluzole, which blocks glutamate release, but what it does is really marginal and extends lifespan by only about 3 months. So it happens that majority of ALS are sporadic. And recall I say that sporadic ALS can be just caused by interplay of multiple genes, stochastic events, and unfortunately, this is the area of ALS that we know very little about. We know very little about how developmental, environmental, or age-related factors contribute to sporadic ALS. But on the good side, we do know a little bit more on familial ALS. And essentially, this can be caused by mutations in a few genes. I've mentioned some earlier. These are SOD1, TDP43, FUS, or a newly identified gene known as C9orf72. So the strategy we're taking in the lab is to derive skin cells from people suffering from ALS, from the patients, to derive induced pluripotent stem cells, or these are cells that can differentiate into any cell type in the body. And we are interested in this case to form motor neurons, which are the neurons that die in ALS. And we want to do that to be able to dissect molecular pathways that are affected in these ALS motor neurons to target them for drug screening approaches. So we have a very robust protocol for deriving motor neurons, both from healthy individuals as well as in this case an ALS patient suffering from the SOD1 mutation. When we stain them for motor neuron markers, ILOT1 and SMI32, what you would appreciate from this slide is that there are no physically or there are no physical differences between healthy and ALS motor neurons. They essentially look identical. But when we start to dissect molecular pathways and ask the question, what is different in these ALS motor neurons? When we looked at metabolic profiles, we saw that the ALS motor neurons are dramatically different from the healthy ones. So the way they utilize energy is very different. We saw that they're using more glycolysis in ALS, more TCA cycle activity, but very, very little of the mitochondrial activity that's going on. So when we do this biochemical assays to validate our gene expression data, what we found was also the same. If you look at these colored lines, they refer to ALS, and the black line refers to healthy motor neurons. What you would appreciate is that the ALS motor neurons use a lot more glycolysis in this extracellular acidification assay, and less of the oxygen consumption assay, which measures oxidative phosphorylation. Not surprisingly, these ALS motor neurons also release less ATP when we did this ATP detection assay. And we were interested to find out what mechanism could contribute to this phenotype. So when we were working in a different project, we came across this piece of data by surprise. So when we were looking at proteome-wide protein acetylation profiles in healthy motor neuron cells versus ALS motor neuron cells, it was surprising that we found two very specific bands or proteins that were hyperacetylated in ALS. And these were identified to be ATP5B and tubulin. Tubulin, we think, is a non-specific event, and we follow up on ATP5B. And when we did this in a genetic form of ALS TDP43 mutation, we saw this. When we looked at a sporadic ALS cell line, we also saw this. So we think this is specific for ALS, but not just to one specific genetic form of ALS. Turns out that ATP5B is a subunit of the ATP synthase, or complex 5 in the mitochondria, which is directly responsible for this ATP generation mechanism. And we think that the hyperacetylation of ATP5B subunit here can lead to metabolic dysfunction. So we went to find out what deacetylates or acetylates ATP5B. And in normal motor neurons, when we treat with a variety of inhibitors against different histone deacetylases or HDACs, we found that the specific inhibition of HDAC6 here, shown by tubistatin A or TUB-A, gives us a hyperacetylated ATP5B. And this also gives rise to a motor neuron selective death, which is ALS-like in nature. So when we look at motor neuron survival when treated with TUB-A against the other inhibitors, what we saw was a motor neuron-specific death without affecting the rest of the cells in the culture. And we wanted to make sure that HDAC6 interacts physically with ATP5B, and what we did here was to use a HDAC6-specific antibody to pull down HDAC6 and every other protein it's interacting with. And true enough, we identify ATP5B, so we are very sure that HDAC6 physically interacts with ATP5B to deacetylate it. So the acetylation of ATP5B is controlled by two classes of enzymes. The first we know is HDAC6, which deacetylates ATP5B, and the other enzyme that catalyzes the reverse reaction are a class of proteins known as lysine acetyltransferases, or CATs. So we know that HDAC6 is probably defective in ALS motor neurons, and the other mode of correcting this disease is to use inhibitors against CATs. So what we have found was that when we treat ALS motor neurons with the inhibitor X, which is essentially one of these CAT inhibitors, we were able to remove the acetylation on ATP5B, thereby leading to better motor neuron survival. And it seems to be specific in that it has no significant effects on healthy motor neurons. So we're quite sure that ATP5B deacetylation is a promising therapeutic target. In summary, I would like to just highlight that we have shown that ALS motor neurons show an altered metabolic profile with defective mitochondria and elevated glycolysis. There is reduced ATP production that's likely due to hyperacetylated ATP5B in both familial as well as sporadic ALS. We've also shown HDAC6 as being critical for ATP5B deacetylation, and the inhibition of this enzyme would cause an ALS-like phenotype. We've also shown with inhibitor X that reversing this ATP5B acetylation can protect ALS motor neurons. This is really a work still in progress, and there are many things that we'll still like to continue doing, and that includes validation in more ALS cell lines, both in sporadic as well as in familial. So I've shown briefly that using one genetic model of ALS and one sporadic line, we were able to achieve this deacetylation with better motor neuron survival. So our challenge next is to repeat this in more cell lines. We would also like to characterize the molecular, metabolic, and functional validation of motor neurons after inhibitor X treatment. Do we see the rescue of ALS phenotype? Importantly, we would also want to test the activity of inhibitor X as well as TAP-A in mice to check if the in vitro results are recapitulated in vitro. And that we would follow with assay development. This could change along the way, but we're thinking along the lines of HDAC6 activity assay, followed by a secondary screen, which might be phenotypic for a motor neuron survival assay. And last of all, ALS mice models would be helpful for our studies in lifespan and behavior studies. So before I finish, I would like to thank the following agencies as well as group of people. Currently, we're working with Thermo Fisher Scientific on a separate project on neuroorganoids. We also have funding from the National Medical Research Council, as well as the Institute. I also like to thank my academic collaborators, Suo Chen in NUS and Bunseng. And last of all, this work wouldn't have been possible without my lab members here. Thank you so much, and I'll be happy to take questions later.
Great. Thank you very much, Xian. So kind of two questions about this. So one thing that seems very promising about this is the fact that it's not only these genetic forms of ALS, but also sporadic forms that seem to be amenable to this mechanism. But I think one key kind of question with all of this is, so in terms of developing a acetyltransferase inhibitor and getting something that's selective and can be used in a chronic manner, because at the moment, most of these acetyltransferase inhibitors are being used in the context of oncology, right, where toxicity isn't such an issue. So have you thought about that, or,
right. Okay, so that's a good question. So in terms of an aged central nervous system, there tends to be, so I'm trying to say that the blood-brain barrier is not so intact. So in the case that, you can have a bioavailable drug that's orally bioavailable drug, and that may cross the blood-brain barrier. In terms of toxicity, I guess, we'll just have to test it out. It's a real
unknown about the research at the moment. Okay. Thank you very much.
Thank you.
Okay, we're staying close to home. We have our next speaker is from NTU, another local institution, Kan-Yi Hu, who is associate professor and assistant chair in the School of Chemical and Biomedical Engineering, and he also has recently returned here to Singapore in the last couple of years. So Kan-Yi is going to be talking to us about a system that he's been developing that looks at kind of molecular imaging afterglow technology. This work at the moment is in press. We won't say at which journal, but I'll leave the rest to Kan-Yi to describe the technology.
Okay, thank you, Andrew. Good afternoon, everyone. So first, I think a little bit about myself. So I did my PhD at National University of Singapore in molecular engineering, and then I went to Stanford for postdoc training for around 3 years. in molecular imaging program. So I came back to Singapore 2 years ago and joined Nanyang Technological University as an associate professor in biomedical engineering department. We are interested in molecular imaging and nanomedicine. Particularly, we want to develop an organic photonic nano platform for different imaging applications, including fluorescence, self-luminescence, photoacoustic, photothermal imaging applications. So in terms of preclinical disease models, we are interested in brain disease, skin disease, drug toxicity, cancer, all different kinds of disease models. But today, I'm going to share with you our recent discovery of a new optical imaging modality, AlphaGlow imaging. So it has extremely higher sensitivity and can be conducted for deep tissue imaging in living animals. So why we are so interested in optical imaging? Okay, if you compare optical imaging with other imaging modalities in clinics such as MRI, PET, ultrasound, X-ray, optical imaging has really higher resolution, can down to nanometer scale, and also in terms of temporal resolution, it's only a little bit lower as compared to ultrasound, but still down to millisecond scale. So they are pretty good. With these properties, actually we can use optical imaging to understand many fundamental processes such as neuroscience. For example, clarity allows us to look at the brain with really higher spatial resolution. Also, optical imaging is very useful for clinical applications. For instance, intraoperative imaging guided surgery because of its higher temporal resolution. Okay, but optical imaging is not perfect. It faces two major challenges. First is tissue transparency. It has really shallow tissue penetration. Okay, that's why it's often only good for surface imaging. Okay, the other issue is tissue autofluorescence. Because of these two major challenges, optical imaging has relatively lower sensitivity, especially in terms of signal to background ratio in deep tissues. Okay, but sensitivity is very important to any kind of imaging modality. You know, with increasing imaging sensitivity, we can minimize the dosage because we need less imaging agents to be injected into human or small animals. Meanwhile, we can increase the ability for early diagnosis. Okay, so talking about early diagnosis, positron emission tomography, PET, has the highest sensitivity, and it's really the golden standard for relatively early diagnosis of cancer in clinics. So the reason is why PET performs so well in this regard. Actually, PET uses radioisotope choices. They can give out signals by themselves. So the imaging process doesn't require any external electromagnetic field to induce the signal to assist the imaging process. So PET imaging also has some issues such as poor spatial resolution as well as very long time scanning. Okay, but the implication here is that if you can get rid of the real-time excitation for optical imaging, it is possible to increase the imaging sensitivity in deep tissue. So luckily, we invited this kind of optical imaging modality, and we call it molecular AlphaGlow imaging. So basically, it contains two steps. First, light irradiation. Basically, you charge the nanoparticle with photo energy, and then you stop light irradiation. You can collect signal released from the nanoparticle. It's called AlphaGlow. Okay, so it has really higher tissue penetration and very high sensitivity because of elimination of tissue autofluorescence. Okay, then I will talk about its mechanism, tissue penetration ability, potential clinical application, and the market need. So we actually find this phenomenon by accident, and this phenomenon is related to this polymer called polyphenylene vinyl, PPV for short. It's a semiconducting polymer, and the mechanism involves the generation of singular oxygen under light irradiation, followed by oxidation of this double bond to form a high energy reactive deoxytane. Okay, this high energy intermediate is not stable and can degrade into aldehyde. Meanwhile, spontaneously emitting long-living luminescence. So the half-life is around 6 minutes, very long. But this polymer is hydrophobic. So we formulated this polymer into water-soluble nanoparticles by using a very simple nanoprecipitation method. And we call this polymer semiconducting polymer nanoparticle, SPANS for short. Okay. Then, because the mechanism involves generation of singular oxygen, so we can dope the nanoparticle with near-infrared photosensitizer to kind of amplify afterglow luminescence efficiency. By doing so, we can also shift the emission to near-infrared range to 780 through an energy relay process, energy transfer process. And because of such a high near-infrared afterglow luminescence, we can detect signal through a living mice. The penetration depth is around 1.7 centimeter, as you can see the signal here. But it's not possible for near-infrared fluorescence, as you can see here. And the signal to background ratio is actually more than 100 times higher as compared to near-infrared fluorescence. Okay? And also, we're looking into biodegradability and the clearance of the nanoparticle. So span, this kind of nanoparticle can be broken down by peroxidase, and then you can see the decrease of absorption, fluorescence, as well as luminescence. So after systemic administration into living mice, the major organ it goes is the liver, as you can see here, but it can be cleared out through biliary pathways within 2 weeks, as you can see here, the decreased fluorescence over time, okay? And then, interestingly, we find such afterglow luminescence is storable. What I mean is, if we irradiate a nanoparticle with light, and then we store the nanoparticles in the freezer, so when we want to do any imaging, we just take the tube out and warm it to the room temperature, and then we can inject into animal for any imaging purpose, such as lymph node mapping, such as tumor imaging, okay? And because of especially higher signal to background ratio, we can actually delineate the tumor in a much rapid way as compared to near-infrared fluorescence. You can see here at 2 hours post-injection, in the afterglow imaging, we can detect tumor, but not possible for fluorescence. So it's a much rapid diagnostic method. And then we can also use this kind of imaging potentially for intraoperative imaging guided surgery, right? In addition to high sensitivity, we can have simplified instruments because we don't need excitation light, right? We can also have improved freedom for surgeon because there is no worry about blocking the incoming light beam during the surgery, right? So this is the potential advantages. And also, we can use those afterglow nanoparticles to detect the biomarkers in different disease settings. For example, we can use these nanoparticles to detect drug-induced hepatotoxicity, right? So while the drugs could be toxic, the toxic potential of a drug is actually determined by detoxification and bioactivation. So traditionally, hepatotoxicity is actually predicted by using in vitro assays to measure protein binding potential, but measuring protein binding potential is really not mechanistically linked to hepatotoxicity and has relatively lower prediction power. In contrast, we can detect a detoxification process by measuring biocells in the liver in real time. So we set out to redesign the nanoparticle by putting such kind of cell cleave quench on the nanoparticle surface to create electron transfer so that the afterglow is quenched at the beginning. And then, in the presence of biocells, the quench is cleaved away, then the afterglow is turned on. So it's an off-on transition process. And we validated our probe in mouse model using a model drug called acetaminophen, APIP, which is a pain reliever and a fever reliever. You can see from these images, for drug-treated mice, the signal is lower as compared to healthy mice, as compared to antioxidant-protected mouse, as well as compared to enzyme inhibitor-treated mice, as shown here. Okay, so our probe really can do real-time drug toxicity detection in living animal, and we also compared our technology with the golden standard histology. As you can see here, the histology here, at 45 minutes after drug treatment, the liver tissue from drug-treated mice shows no difference from healthy mouse. Only at higher dosage, at a longer drug exposure time, 180 minutes, you can detect protein nitration as well as cell death. But if we're looking at the dynamic data of our imaging probe, we can sense that we can detect drug-induced hepatotoxicity within half an hour. So we can actually predict drug-induced hepatotoxicity prior to histological changes. And also we can conduct longitudinal imaging rather than one-time point data collection for histology. And because the signal is closely correlated to the concentration of biomarkers, in this case biocells, we can predict, we can determine the toxic dosage more precisely as compared to histology. Okay? And then we hope that we can introduce this imaging technology prior to call it IND sweet spot in the drug development process, so that we can procure toxic drug from clinical trials and we can save IND cost and deliver safer with safer dosage to the patient. Okay, so both the market for both imaging-guided surgery and the drug toxicity is increasing, and particularly the global toxicology testing market is expected to be around $30 billion by 2090. So we have patented this technology and we are interested in its commercialization. To wrap up my talk, I think off-grown nanoparticles have really good promise for ultra-sensitive intraoperative optical imaging, such as lymph node mapping, tumor imaging, and also they can be served as a very versatile platform for develop smart probes to detect biomarkers in disease settings. And in the future, we want to develop smaller polymers so that they can be cleared out through renal clearance more quickly to solve the toxic issues. And also, we want to incorporate other functionalities into off-grown nanoparticles for different imaging and therapeutic applications. Okay. With that, I'd like to thank my supervisors at Stanford for their kind support, and my collaborators for their help, my group members for their hard work, and also this funding agency for financial support. Thank you very much.
Thank you very much, Kenyi. So, a whole array of applications of this technology. I'm just interested in this last application that you talked about. So is your thought actually that the technology could